All right, is this on? Oh, it is on. Okay, great. Hello, everyone, and good afternoon. My name is Patrick Fox, and I'm the Dean of the Russ College of Engineering and Technology. Thank you for your attendance today for our Russ Prize lecture, featuring Dr. David Walt, the recipient of the 2023 Fritz J. and Dolores H. Russ Prize from the U.S. National Academy of Engineering. Dr. Walt received the 2023 Russ Prize for the development of microwell arrays that have greatly advanced the fields of genomics and proteomics. Let me first provide some background on the Russ Prize. The Fritz J. and Dolores H. Russ Prize was established in 1999 and named after Fritz Russ, an esteemed engineer and founder of Systems Research Laboratories here in Ohio, and his wife Dolores Russ, a longtime supporter and benefactor of the engineering profession. The National Academy of Engineering established the Russ Prize at the request of Ohio University to honor the Russes for their dedication to higher education and the engineering profession. Fritz Russ graduated from Ohio University in 1942 with a Bachelor of Science degree in electrical engineering. The Russ Prize recognizes an outstanding bioengineering achievement in widespread use that improves the human condition. Recognition of these achievements provides visibility such that the public can understand and appreciate the contributions of engineers to our health, well-being, and quality of life. An auxiliary purpose of the Russ Prize is to encourage research collaborations between engineering and the medical and biological professions, which is something we've been talking more and more about here on campus actually lately. Awarded biennially, the recipient receives a $500,000 cash award and a commemorative medallion. The Russ Prize is the top bioengineering award given worldwide, with former awards acknowledging breakthroughs in kidney dialysis, automated DNA sequencing, and LASIK eye surgery procedure. Now I would like to introduce today's honored guest. David R. Walt is the Hans-Georg Weiss Professor of Bioinspired Engineering at the Harvard Medical School, Professor of Pathology at the Brigham and Women's Hospital and Harvard Medical School, a core faculty member of the Weiss Institute at Harvard University, an associate member of the Broad Institute, a Howard Hughes Medical Institute professor, and the co-director of the Mass General Brigham Center for COVID Innovation. He is the scientific founder of Illumina Incorporated and Quanterix Corporation, and has co-founded multiple other startup companies in the life sciences, including Altaview, Arbor Biotechnologies, Sherlock Biosciences, VizGen, and Taurus Biosciences. Now, I don't know about the rest of you, but I thought I was busy. This is something, I gotta say. Dr. Walt has received numerous national and international awards and honors for his fundamental and applied work in the field of optical microwell arrays and single molecules, including the Kabiller Prize in Nanoscience and Nanomedicine in 2021, the ACS, which is American Chemical Society, Catherine H. Hatch Award for Entrepreneurial Success in 2017, the Ralph Adams Award in Bioanalytical Chemistry in 2016, the ACS Gustavus John Esselin Award in 2014, the Analytical Chemistry Spectrochemical Analysis Award in 2013, the Pittsburgh Analytical Chemistry Award in 2013 as well, and the ACS National Award for Creative Innovation in 2010. He is a member of the U.S. National Academy of Engineering and the U.S. National Academy of Medicine, a fellow of the American Academy of Arts and Sciences, American Institute for Medical and Biological Engineering, American Association for the Advancement of Science, and the National Academy of Inventors, and was inducted into the U.S. National Inventors Hall of Fame. Dr. Walt received his Bachelor's of Science degree in chemistry from the University of Michigan and a Ph.D. degree in chemical biology from the State University of New York at Stony Brook. Please join me in welcoming Dr. David Walt to Ohio University for his lecture entitled Discovery, Scale, and Impact. Dr. Walt, the podium is yours.
Thank you. Dean Fox, thank you for that incredible introduction. I have about 5 minutes left to give a talk. Well, thank you for this great honor. Beautiful campus here. I was saying at least this time of year, it sells itself, just the cherry blossoms, and the buildings are amazing resources you have here. So great to be here. And I'd also like to acknowledge my thanks to the Russ family as well. So today I'm going to talk about some discoveries that actually led to the success of those companies. And I want to tell you really about sort of the essentially from bench to bedside here. That's maybe a little subtitle that you could think about. It's really that idea that happens in the laboratory, and then how those ideas end up getting translated into the commercial sector, and eventually, we hope, if possible, they actually make an impact on human health. And so that's going to be really the title here. And so I'm going to tell you two stories, one about the company founding of Illumina, and the other about the company founding of a company called Quanterix, which may not be a household name. I think Illumina is, I think to many of you, at least working in the biological sciences, I think Illumina is a household name, and thank you for your for being a customer. So this starts back in actually the mid-1990s. We were working in the field of optical sensors, and at the time, what we were doing is we were just putting little fluorescent polymer layers on the end of communications fibers, just the same fibers that are brought into your home for cable TV, for data transmission. And what you could do with those is you could do things like put a fluorescent dye that was sensitive to oxygen. You could put that into the bloodstream. You could make a fluorescent layer that was sensitive to, say, an organic vapor that could measure in the ground the leaks from gas tanks. And that was sort of what we were doing. I was, trained to modify surfaces, and so that's what we were doing. And then back in the mid-90s, we started working with this kind of a substrate, which is a imaging optical fiber. It actually has many very, very tiny optical fibers. So these are the light-carrying parts, and then this darker gray region is the part that confines the light into those light-carrying optical fibers, okay? So this is an array. The size of this, you can see, is about, this is 200 microns, so that's about 0.5 millimeter right there. And in that little structure, which is a couple diameters larger than a human hair, at the time, there were about 2,000 of these individual optical fibers. And so a postdoc in the lab, Paul Pantano, who's now on the faculty at University of Texas at Dallas, treated this glass fiber with hydrofluoric acid. And anybody who's ever etched glass, hydrofluoric acid etches glass. That's how glass is etched. And what he found was that the cores of the fibers actually etched a little bit faster than that cladding material, that dark gray material, and it created these microwells right here. And so the way you should think about this is this surface right here at the bottom of this little microwell is actually the end of an optical fiber. And so you could actually shine light into it, and you can interrogate the contents of that individual microwell. But we didn't want that at the time. We were not trying to get this to happen. This was a mistake. This was the opposite of what he was trying to do. What he was trying to do was to create these nanotips. And the reason for that is, at the time, for anybody that was doing any microscopy at the time, there was something called these optical nanoprobes, and you would take one of these little tips here, just a single one, which was made kind of in the same way with a conventional optical fiber. That is, you dip the fiber into hydrofluoric acid, and it would etch to a very sharp tip. You would coat that with gold, punch a little hole in the end, and you'd get a nanometer hole at the end that you could then put onto a surface and scan, and you could get what was called diffraction limit resolution, meaning the light that was coming through that little hole was about 10 or 20 nanometers, and that's about 20 to 30 times smaller than the diffraction limit of light. So you could get images that were very high resolution. So we were trying to do this because then you could do all this in parallel. You wouldn't have to scan a surface like this, you just have to wiggle this array of nanotips. So this we thought was a great idea. And in fact, we accomplished it the year after, but it turns out that this was a transient discovery because then super resolution microscopy came out and all sorts of other advances in technology that made this obsolete. But the microwells turned out to be important. And so this is what I want to talk about is scale. And those of you who work in biology labs, I think you recognize this. This is a microtiter plate. This is what was considered a microwell array back in 1996 when we first developed these microwells. And so here I'm holding one of the microwell arrays that we eventually made 60,000 wells in this little tip. And so you can see the numbers here, 1,000 times as many wells as here, and each of those wells holds a volume of liquid that is 5 billion times smaller than the amount of liquid that's held in one of those microwells. And so particularly the students in the back of the room, and even in the front of the room here, I always want to tell you, when you see something that has that many zeros, either in front of it or behind it, pay attention to it. Because we did not pay attention to those microwells for about a year. So why do we start to pay attention to it? So this is, again, what the architecture looks like. This is pretty close to what I was just holding in my hand in the previous slide. It's about 0.5 millimeter across. It has these 6 fiducial marks here so that if you take an image of any of these wells, you can line them up perfectly. All 60,000 wells are aligned. And each one of those microwells in that array is a very tiny volume and can be addressed by the fiber from the back of the screen there. And so this was an experiment that a graduate student, Carrie Michael, did it in late 1997. And Carrie came into my office. She said, I've got these little latex beads, these plastic beads. What do you think about putting those plastic beads in these wells? And I said, Carrie, go ahead and try it. I don't know, what it's going to be useful for. And she came back a couple days later, having taken these scanning electron micrographs, and all she had done is she had taken a slurry of little plastic beads, latex beads that were about 2.8 microns. The wells themselves are about 2.9 microns. And so she just put a drop on the end of that little optical fiber and then wiped it with a Kim wipe, a piece of Kleenex, and brought this into my office. So each of those wells just self-assembled a bead. Pretty beautiful. We had absolutely no use, but the next year we were working on, remember I told you we were putting fluorescent dyes on the ends of fibers. 1997, 1998, 1996, we actually had published papers where we had put single strands of DNA sequences on the ends of these optical fibers. Just single fiber. So one fiber, one DNA sequence, a second fiber, another DNA sequence. And we actually published a paper in Nature Biotechnology in 1996 describing the first, what we call the DNA microarray. And what it allowed you to do is, if you dip those fibers into a solution containing a gene that was complementary in sequence, we had it figured out so that if things hybridized and made the double-stranded DNA, you would actually get a fluorescent signal from that fiber. And you could actually measure the presence or absence of a gene. And so you could do gene expression using this approach. Well, I was sitting with a colleague who was actually from a commercial company, Beckman Coulter. At the time, they were making oligonucleotides. And this was, for those of you who are working in the field, now you can buy oligonucleotides. They're cheap. You can order them online. You get them the next day. There were only a few labs in the world that were making oligonucleotides back in the mid-90s, and they were expensive. They were very expensive to make. And I was talking to this colleague who was supplying me with these oligonucleotides, and he said, David, I've been talking to some of my genetics colleagues and biology colleagues, and they're telling me that they like your microarray stuff that's where you're able to measure 5 or 6 things at a time, but they're telling me that sometime in the future they're going to want to measure 100 sequences at a time, and maybe in the distant future somebody might want to measure 1,000 sequences at a time. At that instant, I just had an epiphany. And I remembered Carrie's beads in the wells. And I realized that if you put DNA sequences on the beads and you encoded the beads with different fluorescent dyes, you could do what is shown here, which is, this is just a small part of that optical fiber. The entire optical fiber would take most of the front of the room here. But each of these little fluorescent spots here corresponds to a particular DNA sequence that you can measure. And even though there's only 4 colors, this is just one of many different images from this. And so I'm not going to get into the complexities here, but the point is that at the time, there was a company called Affymetrix, which was making what were called gene chips, and the feature size of their gene chips where you could measure a single DNA sequence is outlined with this rectangle right here. But we could put 1,000 beads in that same location. And so, and also, this is completely reconfigurable, because if you want to add another gene, you just put another set of beads in there. And so this idea back in 1997 was really the basis of the company Illumina. And a couple years later, we were already at the scale where we're measuring thousands of genes, even though the colleague had told me that would happen in the very distant future. So I'm going to talk you a little bit through this, because what happened was I gave a talk at Scripps Research in La Jolla, and in the audience, somebody had been clued into this idea. We had patented it, filed the patent, and somebody had wired to a colleague that I was going to be giving a talk, and that this person should come. And after the talk, he came up to me, a guy named Larry Bach, and said we should have breakfast the next day. And literally, that breakfast changed my life. And I think in the end, that collaboration really changed the world from the perspective of being able to bring a transformative technology. So what I want to spend a couple minutes on is really what the company did. It's one thing to be able to measure gene sequences, but what really transformed the whole field of genetics was the idea that this technology was able to measure single base changes called single nucleotide polymorphisms, pronounced SNPs. And this is just to show you that, this person, all the DNA all their DNA is the same, except for at this particular position, in this person they have a G, this person has an A. And so if we go around the audience here, our genes differ. The common variation in our genes is about 1 in 1,000. We have 3 billion bases in our DNA, and about 3 million of those are different common SNPs, the common differences between any two of us in the room. But it's not that simple, because if we go to a particular site of DNA, most of our DNA is common, but at this particular site, it may be that 20% of you have an A at that position and 80% of you have a G. And if you go to another position down the road here, it might be 50-50. And in another position, it might be 70-30. And so that's what genetic variation is. It's those 1 in 1,000 particular positions. And so Illumina decided that this was going to be the way that they were going to use this technology, was to measure SNPs. And that became the basis for what is now called genome-wide association studies. You can take a population, and one of the first populations that they did was to look at diabetics. And you could look at diabetic patients, you could look at healthy patients, and you could see where the genetic variations were. And by that, you now know which genes are contributing, because you could find the locations along the DNA. You could figure out which genes were responsible for that disease. Some diseases are simple, like sickle cell, just a single base change. Other diseases like diabetes are more complicated, could be hundreds or thousands of differences that actually lead to different manifestations of the disease. And so at the time that Illumina decided to go into this business, in order to make a measurement of one particular site, one particular SNP, it cost $2 to do that, okay? So if I wanted to do a genome-wide association study with 1,000 people, and let's say I wanted to measure 1,000 SNPs, well, 2 times 1,000 times 1,000, that's $2 million. That's a lot. It's a lot of money. Can't do it. So the first product that was introduced was essentially what my lab had developed. It was called the array matrix. It was an array of 96 optical fibers with 60,000 microwells, with 60,000 beads. Each of those fibers was able to do 1,536 SNPs. Now, why couldn't you do 60,000 SNPs? Well, I think most of you are engineers. You understand what the Poisson distribution is. It means that if you want to make sure that every one of those genes is represented on that array, and you have 60,000, you have to make sure you have between 20 and 30 copies to ensure that there's at least one bead in that array that has that particular gene. Otherwise, you're going to leave a bunch of them out. And so what we did is we made it configurable. If you wanted to measure 1,536 genes in 96 patients, you just put the 96 patient samples in a microtiter plate, you dipped it in. If instead you wanted to measure 1,536 times 96, you would just put the same patient sample in all 96 wells and dip it in, and then you could do a few 100,000 genes in that particular format. Well, the technology rapidly outgrew that, because what the company did is they started making microwell arrays in silicon, just etching silicon, because you could make this in a planar fashion. You could make it much more dense. And back in 2021, remember I said 1,000 genes in the far future? Well, 20 years later, they're measuring 28 million SNPs per individual. And how much does it cost? Well, I'll tell you that in a minute. But this particular chip does 8 human samples, 28 million markers per chip. Now, you say, why do you need 28 million? Well, over time, we've discovered that there are not just 3 million common SNPs, but there's lots of rare ones as well that lead to other, more other aspects of human genetic variation. Okay, so I'll just show you a couple of pretty pictures. These are what these things look like. The beads go into the wells. They snuggle in there just the way Carrie Michael did. Pretty remarkable. You can't get those things out. And they have nice diffraction patterns here. And so this is the price. Okay, went from $2 to what's that? 400,000 or 4 millionth of a dollar, something like that. Okay. Incredible. Really, never in history had technology advanced this fast. A lot of zeros there. And that's what enables these. You can do 28 million SNPs for $79. So this is what these are used for. How many have had 23andMe done? How many admit, you're the only one that's admitting you did 23andMe. I've done it too. But this is recreational genomics, I call it, because you find out interesting things about. When I had this, I was one of the first 100 customers of 23andMe. I was on the Illumina board for 18 years, so the board got a discount. The most useful information that I got from this, because at some point you learn about whether you have wet or dry earwax, it will tell you about your different gives you does give you your ancestry. But the most interesting thing that I learned from this is that I am a fast caffeine metabolizer. Now, before I had 23andMe done, I would never drink coffee after 5 o'clock at night, but because I was up at night. But we found out that my wife was a slow caffeine metabolizer, and so she's just going, David, are you up? Are you up? And so I go, yeah, I can't sleep. Must be that coffee. So now I can drink coffee after dinner with impunity. It's also the technology that's used for Ancestry.com. This is actually the biggest. About 20 million people have had their ancestry done with this technology. Now, most people think that this is done with sequencing, but it's not. It's done with these microwell arrays because it's cheaper to do it. It's still much cheaper. And in fact, Illumina sells millions and millions of microarrays and many fewer full sequences every year still. But back in 2006, we acquired a chemistry technology out of University of Cambridge in the UK, and started to and introduced some sequencing technology. And just to put this into scale, the Human Genome Project took 13 years. It was 40 large institutions around the world. They divided each chromosome to different institutions. You could on these gels, you could do 1,000 bases at a time. It took 13 years. The technology that was referred to, the automated sequencing, was actually developed by Lee Hood at Caltech, and he developed the first automated sequencer at this particular time during these years. But after the Human Genome Project was done, the first next-gen sequencer was introduced by Illumina, and it could do higher throughput. But the whole sequence, the whole Human Genome Project cost $3.8 billion, took 13 years. Thousands of people worked on it. Today, one instrument, a NovaSeq, does 16 trillion bases per day, takes less than one person. It does 200 genomes, and it does that at 30x coverage. It means that you resequence each person 30 times to make sure that it's a perfect genome. This was an imperfect genome. They did it only 8 to 9 times, and it cost $200. Now, the entire Human Genome Project could be done today in 7 minutes. 19 million times cheaper. One instrument is 1 million times faster than 40 labs combined. Again, just remarkable advances in technology enabled by engineering. And the reason I'm covering this is not just to inform you about sequencing, is that this was the end of the Human Genome Project. The technology had actually developed to the point where at the end of the Human Genome Project, if you wanted to do it again, it wouldn't cost you $3.8 billion. It would only cost you $100 million. But here, Illumina introduced their first next-gen sequencer, and so prices began to plummet, but they got stuck at $10,000. And they got stuck for quite a while because the density of the features on the sequencing machine could not get small enough until the company decided to do sequencing using microwell arrays. So they took the microwell arrays from their microarray business, and they put their sequencing chemistry onto the microwell array technology that we had developed, and that's when we've achieved the first $1,000 genome, which was really this was a milestone in our ability to sequence, because at that price point, it almost became a commodity to start sequencing, and today, as of last year, it went down to $200. This is NIH just stopped updating these. I don't know why they stopped, but I think it was just because they got tired of just keeping it at $800. But it went down to $200 because the patents ran out, and so now former Illumina employees went and started competitive companies and priced it at $200, and so Illumina had to match that, and so now it's $200 for a human genome. Okay, so this is, some people stop here with impact. You've created a company, $30 billion company. At one point, it was $90 billion, so still pretty good, but not as good as it used to be. Revenue is $4.7 billion, and all sorts of awards that they've gotten, but really, that's not where the impact starts. This is where things really started to change the way I thought about translational medicine and taking technologies into the private sector. It's when, it's these two cases. So this particular woman suffers from a genetic disease called Charcot-Marie-Tooth syndrome. It's a degenerative disease. Her grandfather had it. She has it. And this couple did in vitro fertilization. So, well, I don't have to talk about, it's been in the news, but it's woman's eggs, man's sperm, fertilized eggs, And what they used, this was the microarray technology. You could genotype the early stage embryos and choose the embryo that did not inherit the mutation, and then implant that embryo into the woman. And now this beautiful baby Lucas was born, who no longer is a carrier of the disease. And so that genetic disease has been taken out of the family line. No genetic manipulation, no CRISPR or anything. This woman was an older mother, and at the time, again using the microarray technology, they developed a method for looking at fetal DNA in maternal blood. And this was the first genetic test that could be done while the baby was still in utero for trisomy 21, Down syndrome. Prior to this, they would do amniocentesis, which was potentially, could potentially lead to spontaneous abortion. So you could take the blood, you do a ratio between the intensity of the mother's DNA to the baby's DNA. If it's 1-to-1, it means that there's 2 chromosomes from the baby, 2 chromosomes from the mother. If it's 1.5, it means trisomy, meaning 3 chromosomes from the baby. So they did that, and they found, so that was the first genetic test that was introduced, and they found in this woman there were some anomalies with the genetic analysis. And so they, at the time, back in the mid-15s, they reflexed to amniocentesis. And they tested, and it was fine. It turned out that she had circulating cancer DNA. And so they waited for a few more weeks, induced labor, started her on chemotherapy, and everything worked out well. But this was really the first time that this was discovered, and it was accidental. It was discovered as a consequence of running this non-invasive prenatal testing. And so now this is a very common thing. So this is really, to me, this was what all of a sudden struck me, that now this is not just an economic impact, it's really human health impact. So I'm going to tell you a second story here about a second technology that uses the microwell arrays, and I want to just be cognizant of the time here. So again, one of the things that we just completely didn't recognize is that the volumes here were really tiny. And I told you before it was 5 billion times smaller volume than the microwell arrays, than the microtiter plates. But the volume in here, contained in this little microwell, is 30 femtoliters. So that's 30 times 10 to the minus 15th liters. And so at a lab meeting, I asked my lab, my group, I said, let's figure out what the volume is. We figured it out. And I said, well, how many molecules would it take if you put them in there? How many fluorescent molecules would it take to be able to detect them with the naked eye? And we calculated it turned out to be 100 molecules. Not very many, because if you put a small number of molecules in a very tiny volume, you get a locally high concentration. And so what do? We put enzyme molecules into these wells at a very dilute concentration with a substrate that is a molecule that the enzyme catalyzes to form a red fluorescent product. And so we then sealed that with a little silicon rubber, and Here's in the absence of any enzyme. And then in a dilute solution of enzyme, you get an occasional well that lights up. And even more dilute, you get fewer but still detectable fluorescent spots because that enzyme is catalyzing the turnover of that starting material into the fluorescent product at a rate of about 400 molecules a second. So you just have to wait a few seconds and you get this. So we said, well, what can we do with this technology? And so, well, we've already solved the measurement of these things. What about proteins? And so at the time that we came up with this idea, we knew we could measure about 320 proteins in the blood. High concentration protein like albumin, low concentration of a particular protein would be something like one of the cytokines like TNF-alpha or interleukin-6, things that cause inflammation or immune responses. And so we said, what if we could access all those proteins that we know have to be there because there's over 20,000 genes and there's thousands of proteins that are expressed by those genes, so there has to be thousands of proteins in the blood that we can't measure. And so we said, well, what if we could digitize protein measurements by counting protein molecules instead of using this kind of approach where we just kind of look at the intensity, which is the conventional technology called ELISA technology. It's immunoassay technology that's used by just looking at the intensity of the color. You've all run your COVID test. That is exactly this kind of test. This is the sophisticated quantitative version that's run in a microtiter plate. But we said, if I asked you what's the concentration here, you couldn't tell me unless you had some way to compare it. Here, this is an absolute measurement. I can tell you there's 4 molecules here. And so this is not a technical talk. I'm not going to tell you exactly how we do this, but we published a paper in Nature Biotech in 2010, and we started a company around this called Quanterix, which this particular instrument can do four 96 well plates of human clinical samples in 5 hours. This is the more manual version of it. But the result of this is that, again, we've translated this technology into the commercial sector where it can actually be used to do important things. And I'll just talk you through that in a minute. This has a slightly different format. The microwells are right here on these little squares here at the end of this plastic disc. Does anybody recognize this plastic disc? I see the folks with some gray hair shaking their heads. This is actually made by Sony because they were not selling DVDs and Blu-rays because everybody's going to MP3, MP4 format. And so they decided to get into the life sciences business. And this was actually the first product that they made for life sciences. So there's 24 microchannels here that deliver beads to those wells, and those beads actually have the assay chemistry on them, so that the on beads means that there's a single protein molecule, and the off beads means that there's nothing there. And that's that's how you digitize things. So here's what these things look like. Really excellent. She just came out with a new book on how to visualize science, so I encourage you to look for that. I think you can get it on Amazon. I don't get any royalties from that. So this is these are smaller beads. They fit into the wells, and then they just give you a signal that looks like this. if there is a protein molecule. And so all you do is you count the number of on beads to the number of off beads, and you get a quantitative measure of the protein. The advantage, and this is what the beads look like. This is a bigger field. The advantage of this is that you can get 1,000 to 10,000 times more sensitive, lower concentrations. And so now we can access that iceberg that's under the surface. So this company, a little bit further behind, it's located just outside of Boston. Last year, they sold 120 million in revenue. It's all only for research use at this point. But I want to show you some of the clinical studies that are being done so you can kind of get excited about what the potential for being able to measure things at ultra-sensitive levels means. And I think this is going to really have a big impact on healthcare in the future. So this was one of the first things that we studied was men who've had prostate cancer and have had surgery, a radical prostatectomy. There's a molecule called PSA, prostate-specific antigen. It's a protein that's put out by the prostate gland. And usually in men who have prostate cancer, their PSA is crazy high. And then after surgery, what happens is they no longer have much prostate tissue left, and their PSA level plummets to below the detectable level. So this was existing test right here. But in many of these men, either the surgery was, they left a residual tumor because the surgical resection was incomplete, or the tumor had metastasized prior to the surgery. And so men have to come in every 6 months to get their PSA level measured. And if the tumor grows, what happens is eventually it's producing enough PSA that it goes above the detection limit for the existing tests. And they say, oh, you have biochemical recurrence. So what we did is we took men who were 5 years out from their surgery who were undetectable. This level is the ultra-sensitive test. And we could quantify every single one of those patients. And you can see this is our detection limit here. Compare that to this blue bar here. It's about 3,000 times more sensitive with exactly the same reagents. It's just digitizing the signal. And so we were able to actually predict from this from a retrospective set of samples, that is samples that had been collected periodically, that we could predict within 6 months after their surgery by measuring their level of PSA. If it was above 3 picograms per mil, they had a 65% chance of recurring. If they were below 3 picograms per mil, they were essentially cured. So they don't have to wait 6 months and come in every 6 months and have things unknown. So we're now applying this to lots of different things, neurodegenerative diseases, different cancers, infectious diseases. I'm going to talk you through very quickly 2 little stories here. Breast cancer. So 1,000 women who come in for mammograms, 900 of them are okay. 100 of them have to be called back for a further diagnostic workup, ultrasound. Of those 100, 80 of them come back. Some of them have to have their mammogram redone. 20 of them have to have a biopsy. Of those 20, 4 end up getting diagnosed with cancer, 16 are okay. That's pretty awful, right? Look at those odds. Let's forget about this. 4 out of 100 that get called back have cancer. It's good that it's a small number, don't get me wrong, but 96 of them have to undergo the mental anguish, not knowing, having to have further intervention. So we've assembled a team. We measure 24 proteins. you can look at those. I can't make any heads or tails of anything. But if you put this through a classification algorithm now called artificial intelligence, what you get with those 24 is you get a accuracy of about 95% just from a blood test by measuring these very small, low concentrations of these proteins. You can't, if you look at those, you can't tell the difference between a healthy and a cancer patient, but the algorithm can be trained to recognize that. Even 4 biomarkers give you about an 87% accuracy, and we think, as we were talking at lunch, that something like this could be really beneficial in low and middle income countries where this test is $2. And we can even subtype. We can tell whether it's hormone receptive, hormone positive, triple negative, et cetera, with the same markers. We've been involved. You heard I've been involved as director of the COVID Innovation Center at Mass General Brigham. We've been involved with COVID, and we've had lots of done lots of work in this area. But the one I want to spend 1 minute on today is this post-acute sequelae of COVID, also called long COVID, which Americans, the estimate is somewhere between 15 and 25 million of U.S. population are still suffering from some post-acute symptoms of their disease. And what we're looking for, we just did a whole sweep of lots of different proteins. And what we found, to our surprise, is that in a preponderance of these individuals, of patients that have long COVID, there is the presence of that spike protein, that thing that sticks out of the coronavirus, the full spike protein circulating in their blood up to, now we know, up to 2 years after they've had their infection. What does this mean? It means that there's still virus that's somewhere hiding in their bodies. And so we're now, I'm a co-principal investigator on a clinical study called VITAL, which is looking at whether you can use a long duration administration of an antiviral, Paxlovid, doing 25 days and 15 days. Now, why the multiple of 5? They come in 5 packs, and Pfizer does not want to repackage that. But 15, 25-day regimen, we're about to get our first readout from this. The hope is that, what we're hoping is that patients who have the spike protein present, that spike protein is going to go to 0, go to baseline. It means that the virus will have been wiped out, and those individuals will literally recover from their long COVID symptoms. Patients who do not have that spike protein probably have long COVID due to some other cause, which could be just organ or tissue damage that occurred during the acute phase of infection. And so this would, if this reads out the way we think it's going to read out, then the presence of that protein that's measured at ultra-sensitive levels will be the eligibility criterion for getting the antiviral. So if you get a blood test, you have the spike protein, you get a 15 or 25-day regimen of Paxlovid. So this is across the country right now. So I mentioned a few of the people that, these are the people, not everybody that's been through my lab. I've had a few hundred people in my career, but these are the people who actually were responsible for the work that I talked about today. Lots of incredible support from foundations, government, and benefactors. This is our retreat last April, just about a year ago. So this is the present laboratory, a rich group of a few undergraduates, a few graduate students, many postdocs, 6 clinical fellows, which really helped drive some of the clinical work in the lab. So again, thank you for the honor of being able to present the Russ lecture here, and it's been great to visit. Happy to answer questions now or at the cocktail reception afterwards. So thanks very much.
Hello.
If you have any questions for Dr. Walt, please raise your hand and we can bring the microphone to you.
So, Dr. Walt, what's the practical limitation of being able to solve many of the inherited genetic problems like the lady did on the one case where you were able to identify it and eliminate it from?
Yeah, well, it's a great question. You know, we're sort of at the front end, which is the detection and diagnosis. I think, you know, just being able, you know, like for these neurodegenerative diseases, just being able to diagnose early stage neurodegenerative disease is insufficient if you don't have an effective treatment. But I would say that the hope is that if you now have markers that can differentiate between healthy individuals and disease individuals, and you can administer a drug and you can see that the trajectory is from a higher level of disease state to one that's closer to a healthy individual, I think technologies like this will help accelerate the introduction of drugs. And I think there's wide acceptance now, even by the FDA, that being able to have informative biomarkers that can predict both presence or absence of disease, but also therapeutic efficacy is going to be essential for the precision medicine that we hope is going to happen over the next decade or so. I love questions from students. Come on, you must have questions.
Great talk. That was really, really good. Appreciate you coming here and giving it. I had a question about your last last part of the talk. When you did the thing with the people with long COVID, why did you check at the protein level and not the PCR?
So we did check with PCR. We threw the book at these samples. You know, we measured cytokines. We measured antibodies. Every one of the long COVID patients, they all are negative for nasal swabs, so they have no upper respiratory infection. Some of them have RNA, primarily those individuals who have GI symptoms, you know, because they think, you know, the GI tract can be a reservoir. But in most cases, the nucleic acids, particularly in the bloodstream, there's nucleases that just get rid of them. So the only thing that's left is this residual protein.
Does a lot of them have cytokines? Or is it
No, there are cytokines, and there's some elevation of inflammatory cytokines like IL-6, but most of them, you can't really tell from their cytokine measurement. The problem with cytokines is that baseline measurements of baseline levels of cytokines can differ by 2 or 3 logs. So, sure.
Yes. Thank you, David, and congratulations on the Russ Prize, and what a remarkable career, and it's still going on. Could you mention to the students here, the undergrads and early graduate students, some of the thoughts you had when you were in graduate school? I know you mentioned at one time to me today, you said, I'm not doing this anymore. Could you mention some of that background?
Yeah. Well, most people don't know this. I was actually trained as a synthetic organic chemist. And from my talk, you probably would never guess. I was not trained as an engineer. I've done engineering more than half my career just because of my involvement in technologies. But after I got my Ph.D. in organic chemistry, as I was saying earlier, I said, I don't want to do this again. This is not something I want to spend my life doing. And I found something that I actually decided that I would go learn how to use organic chemistry to attach things to surfaces. So I didn't abandon organic chemistry, I abandoned synthetic organic chemistry. I used that tool that I had learned to apply it to something that I thought was a more interesting problem. And it was kind of a big change, it was a risk. And I've pivoted multiple times even in my academic career. We used to work on something called an artificial nose. It was being able to make an array of sensors that recapitulated the architecture of the olfactory system, and we could sniff explosives and do all sorts of interesting things with it. And I had oodles of DARPA funding for this. And at some point, after about 6 or 7 years working on it, I probably could have done it the rest of my career. I said, I don't want to work on this anymore. We've done like 90% of everything that is ever going to get done. And I just said, I'm going to do something else. And this was right around the time that we did the single molecule work. And I said, I'm not going to renew my NIH grants for the genetic stuff. I'm going to move to proteins. It was a big risk. But, the only way you really can evolve as a scientist, engineer, whatever, is to take those risks and to be confident in your ability to do something different and equally well as something that you did before. It's not as if you have to learn how to do research again. It's just you have to learn the specifics of how to do. You have to learn the lingo of that new field. You have to learn the techniques of that new field and apply your capabilities and skills to that. So,
So you kind of talked a lot about like the human applications for your technology. Has there been many applications as part of as far as like plant science and like new variety, like making and all that kind of stuff?
We don't work with plants, but we do have a number of studies with animals like mouse models, for example, where, so for example, one project that we're doing right now with a colleague in a collaboration is we can look at the cytokine content of mouse poop when the microbiome is perturbed with various drugs or in different ways and manipulated. Because the method is so sensitive, it needs really tiny amounts of biological specimen to be able to make those measurements. So, nothing with plants, but we do work with other kinds of animal systems.
Thanks everybody for coming out this afternoon. I really appreciate your time and attention. Let's thank Dr. Walt one more time. And there'll be a reception outside immediately following. Thank you. Fascinating lecture.